All right, folks, now let's get into the gross anatomy of skeletal muscle. That's going to be our focus for the rest of the entire lecture, okay? And that means we have to understand how the organization of the muscle actually presents. Now, let's get ourselves oriented here. We do have the ball and socket joint here, and this is going to be our humerus, our upper arm bone, okay? We see that we have an attachment to this bone and this piece of muscle, and that attachment is called a tendon. Tendon is going to attach muscle and bone, okay? What else do we see? We see a large hunk of muscle, and then we see that in this hunk of muscle, we pulled out another piece of muscle. And then in that hunk of muscle, we pulled out another piece of muscle. And then as we go along, we're going to put out this last piece of muscle and then go down to the microscopic level. That's the whole goal. But I kind of want to get our brains primed, so I'm going to go back just so I can have extra space here. Let me go ahead and see if I can click on this. Okay. But let's go ahead and just have me rewrite this here. And of course, it's not going to look perfect. But this is our humerus. We have that tendon that's attaching muscle to bone, and really it's just all the muscle fibers coming together, right? And then we have this hunk of muscle, excuse me. We had a hunk of muscle, and all these bundled in. Well, let's go ahead and talk, and then of course I'll give you all the information after. First of all, this tendon is going to attach to the humerus, which is what we see. But what else do we see? We see that we have this whole muscle as well. And surrounding this whole muscle is connective tissue. Okay? So this connective tissue is called epimysium. Epi just means that you're on the surface or outside of something. So it's on the outside of the entire hunk of muscle. And mysium means that we're talking about connective tissue in relation to the muscle itself. Sometimes you'll see myo or mysi. That's going to help us relate it to the muscle. Now, what else did we notice? Of course, it was very quick. But we also had these bundles that when we took a cross-section of that huge hunk of muscle, we also saw bundles within it, okay? And these bundles are also surrounded by their own connective tissue. That connective tissue is called perimysium. So you're thinking of perimysium, thinking it's surrounding something, okay? But what do we also notice? We also notice that each one of these, let me go ahead and get a different color. There was also, in each one of these, there's also bundles. So we have bundles here. I'm just going to put a couple just real quick. Bundles here, and bundles here, right? And I can take away the green here, 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 and here. Just so we can distinguish that these bundles here have their own connective tissue as well. And these bundles are called fascicles. So we have the muscle, and the muscle is going to be, when you take a cross section, you'll be able to see fascicles, and they're surrounded by perimysium. And then, if you go ahead and pull out one of these little purple ones here, pull out one of them, they're also going to have and be bundles of something else. So it's like one of those situations where you have those little Russian dolls that just more and more and more pop out. And guess what? Each one of these is going to be our muscle fiber, also called our muscle cells, okay? And they're surrounded by their own mysium as well, and it's going to be called endomysium, okay? Sometimes people say mysium, I say mysium. Most of the time I hear it mysium, but again, choose whatever connecting you do. So you see how we have this cross-section of muscle. It's made of a bunch of fascicles. Those fascicles are made of a bunch of muscle fiber cells. And then guess what? Yep, keep going. Let me try to get another maybe like, now I'll just erase the little dots, and I'll put little dots here. And if we pull out these little dots, these are actually called myofibrils. And these myofibrils are made up of these contractile units, our muscles contract, called sarcomeres. And those sarcomeres, keep going, and this is the last little bit, are made of myofilaments. Okay? And it's the sarcomeres that allows the muscles to contract. So we'll be going from the larger gross anatomy down to the myofibrils, which again, they're surrounded as well with different, it's not going to be connective tissue, it's actually going to be a membrane now. And they're going to be surrounded with what's called the sarcolemma. Sarco means muscle, another form of muscle. Lemma means flesh. So it's basically a flesh sheet. Okay? That's what they're going to be surrounded by, flesh sheets. Or you can say a flesh cover. The sarco really means flesh. I just want to write that out there. And lemma is like a cover or like a sheet. So you can say it's a flesh cover. We think of flesh, we think of muscle. Okay? So that's a lot going on. And I just want to prime our brains since we're going to get into details of this. So that means you're looking at the muscle. And that muscle is made of fascicles. And those fascicles are made of muscle cells. And those muscle cells are made of, let me go ahead and get another color. Those muscle cells are going to be made of myofibrils. And those myofibrils are made up of sarcomeres. And these sarcomeres have very specific proteins called myofilaments that help us contract our muscle. So we have this in mind. Now let's get into it. Okay? So muscles are comprised of muscle fibers, or muscle cells, nerves, blood vessels, and connective tissue, that perimysium, endomysium, and epimysium that we saw before. I understand you might be like, well, of course it's made of muscle cells, and you just showed me a little bit about the connective tissue, but why nerves and blood vessels? Well, again, for our muscles to be able to work, it's going to have to be able to do what? Use oxygen. How do we need oxygen? Well, go back to cellular respiration. Okay, cellular respiration is going to use glucose and oxygen to form what? Water, CO2, and ATP, or the energy source. So in order for us to get oxygen, we need blood vessels. And so you'll see the blue here or the red here are going to be those blood vessels. Why would we need nerves? Remember we talked about excitability, the ability to transmit stimuli from muscle tissue and also nervous tissue, giving that signal through the axon terminal. It's just like another synaptic cleft that we get to talk about as well. Well, that's where the nerves are important for, right? So each muscle fiber needs the ability to have a nervous system muscle connection so that we can actually have contraction happen, right? So that's the reason why nerves are important. And connective tissue, again, is surrounding each of the pieces of the skeletal muscle as far as growth anatomy. And also, all of these muscle fibers and everything come together to form the tendon that gets attached to the bone. So muscles are made of many wrapped bundles. That's one thing we can talk about. So let's talk about the first one, which is going to be this bundle here. Muscle fibers, or again, muscle cells. They're going to be long, multinucleated. So we know they're multinucleated. We know they're striated. But we also need to get more details and say, hey, these are long. That means if you have one muscle fiber, is going to be a length of whatever muscle you have here. So let's say this is our biceps brachii. The muscle fiber will be as long as the length of this entire muscle. Okay? So if you had a muscle, and I'm talking about biceps brachii because it does have this spindle shape, the length of the fiber will be all the way to the end of that muscle. Okay? Same length. And this is surrounded by endomysium. Why is it endomysium? Endo means within. So it really is the muscle cells that are going to be within this entire muscle. And so that's where you're getting that connection there. Okay? Then we have fascicles. Fascicles are made of bundles of muscle fibers or bundles of muscle cells. And this is a fascicle here. You'll see, we see all the bundles of muscle fibers here. And as you can see, you might be like, okay, what's all this white stuff here? Well, it has to be endomysium because we're surrounded. There's a bunch of bundles of muscle fibers. We know they're surrounded by endomysium, so then that's endomysium. But we also know that the fascicle itself is surrounded by its own connective tissue, and that's going to be called Perimysium. Okay? So you have this bundle of muscle fibers surrounded by perimysium, and then those are also bundles because the muscles themselves are bundles of fascicles. That's not a D, that is a C. There we go. So fascicles are made of bundles of muscle fibers, and then muscle is made of bundles of fascicles, okay? And this is going to be surrounded by epimysium. So here's all the epimysium right here. And you might be like, what about the perimysium for the fascicles? Well, let's talk about that. You see all these fascicles, bundles that we see here? All the white surrounding it is going to be perimysium. So think about this. When you get a good old chunk of meat and you cut it, say you have a steak or something like that, I don't know, but you cut the meat and you see all this kind of marbling in the meat. Well, what you're really seeing is perimysium, okay, because of the fascicle bundles. And sometimes, depending on how fancy the meat is, you can actually end up seeing sometimes endomysium as well. Okay? So, muscle fibers, nerves, blood vessels, and connective tissue all come together to help build the structure of the skeletal muscle. But that's not the only thing. We still have those tendons, right? And the tendons are going to be like rope-like to help connect things to bone. And not only is it just connecting to bone, it also can connect to other muscles, cartilage. I mean, it's connecting to other things in the body. But what we normally see is muscle directly connecting to bone is going to be a tendon. And also, if you think about your abs don't have tendons like we normally see in this rope-like thing. It has more sheets of connective tissue called aponeurosis. And so if you think about it, you're like, okay, do we have a direct connection to the bone or do we have an indirect connection? When you're talking about tendons, this is an indirect connection. It's most likely you won't see endomysium that's connecting to a tendon. You'll probably definitely see perimysium and epimysium that makes up that tendon, right? Although the different connective tissue come together down to create that tendon. And that's an indirect connection to bone because you need a tendon in order to attach. Same thing with the sheets like aponeuroses. That's going to be an indirect connection to, again, the bones around it or cartilage or other muscle around it. So then we do have direct connection. Direct connection will mean that the epimysium, you see here, is going to fuse with the, maybe the periosteum that is surrounding the bone. If you have a direct connection like that, then yes, that can happen as well. So, or even like the perichondrium cartilage. I think that if you're thinking of connection directly to cartilage or bone, you can definitely have your epimysium, again, attached directly to the periosteum of the bone or the perichondrium of the cartilage. Okay, folks? Now let's go ahead and get into some more microscopic, and this is going to be our first kind of taste of the microscopic aspects, because when we start getting into how muscles contract in one theory, which is the sliding filament theory, then you'll basically be able to see, oh, okay, now I understand the microscopic pieces, and then I'm going to keep going with that. What do we know? We know that a muscle fiber must shorten to contract. Okay, that's what we're going to be saying that we know. A muscle fiber must shorten to contract. And so we're talking from going from the muscle to the fascicles to the muscle fibers. Now we got to talk about the other piece, the little pieces, the myofibrils, which are made of sarcomeres, which have myofilaments in them. Okay, so this is what we have pictured here. We see some endomysium here, and it's sort of peeled back. Okay, that endomysium looks like it's kind of peeled back. And what we're revealing is what's called the plasma membrane, or the membrane of our myofibers. And that membrane is called the sarcolemma. And so remember, let's go ahead and get ourselves oriented here. We have muscle cells, so we're not worried about this now. We're worried about the muscle cells, and we're worried about all of this. So muscle cells are made of myofibrils, and instead of connective tissue surrounding it, we have the sarcolemma surrounding it, which means flesh sheet, okay? And those myofibrils are made of sarcomeres, which are like the fundamental units of contraction, the things that actually shorten in order for the muscle to shorten, and proteins called myofilaments. So sometimes you'll see folks say, oh, myofibrils are made of myofilaments. Well, that is true as well, because myofibrils are made of sarcomeres, which have myofilaments in them, right? So we're going to be focusing on the myofibrils, well, the muscle cell pulled back the endomysium, then the myofibrils, pull out the myofibril, then talk about the sarcomeres and the myofilaments within the myofibrils. That's where we're at here. So, of course, we're at more of a microscopic level. Okay? And so, again, The sarcolemma is the plasma membrane wrapping the myofibrils. You also see like these little pink stripes, okay? Those are going to be the T tubules, and we'll talk about their importance next, okay? So the T tubules are going to be membrane, and usually they're like tunnels, which you can also fancy it up and call them canals, that reach deep into the myofibril. So you might be saying, well, why do we need to reach so deep into the myofibril? Well, we need to be able to contract. Remember, we're trying to get down to the microscopic piece right here, which is the sarcomere that actually allows contraction. So in order to do that, we need something that's going to be able to reach that sarcomere. And just surrounding it's not going to be enough. So it's basically like you have in a bunch of rings, little canals surrounding that myofibril as well. So we have that sarcolemma, then we have our T tubules. It's going to be, again, little canals that allow something to be able to be transported to those myofibril so that we can have a contraction. But before we start adding those details, let's continue. Another thing you notice, because I see our T tubules here, so let's go ahead and write those in pink. Boom, boom, boom. And then another thing I noticed is all this stuff in blue. So it's like here, I see it in blue, I see it in blue here, I see it in blue here, I see it blue down here. That's also surrounding it. So all this blue stuff on our myofibril. And you're like, what is that? Well, that is sarcoplasmic reticulum. We know endoplasmic reticulum when it came to just understanding cells, but when we're talking about muscle cells, there's a specific type of endoplasmic reticulum called sarcoplasmic reticulum. Again, sarco means flesh. We're thinking of muscles here. And this is going to surround the myofibril. And one thing it does, one of the big things it does, is store and release calcium ions. So let's talk about why this is important. We have an axon terminal that is going to be sending an electrochemical signal, right? And that electrochemical signal needs to be able to get to the sarcolemma or the plasma membrane here. So what it's going to do is going to release neurotransmitters. Those neurotransmitters are going to hit a receptor on the actual sarcolemma. But if we stay at the sarcolemma, the signal is not going to get down into the myofibril. So we have to make sure that electrochemical signal gets down into the myofibril. So that signal is going to move down the sarcolemma and is going to reach the T tubules. So once it reaches the T tubules, the signal now can get closer to the myofibril, right? And guess what the T tubules do? They're going to have the ability to get deeper into the muscle fiber and stimulate that sarcoplasmic reticulum. So all this blue piece here is going to now get stimulated. Once it's stimulated, what does it do? It stores and releases calcium ion. And it's those calcium ions that gets the contraction going. And we'll talk about this next in the sliding filament theory. Those calcium ions are going to attach to one protein called troponin. That troponin is going to allow the actin filaments, the proteins, to move so the myosin filaments, which we'll talk about next as well, can actually bind, thereby pulling on the muscle and causing the sarcomere to get short. It's not really pulling on the muscle, it's really pulling on the actin to allow the sarcomere to get short. I'm going to talk about those details as we go along, but I want to bring that up just to prime my brain. So these myofibrils are long rod-shaped organelles within the actual muscle fiber, and they contain myofilament. These myofilaments are nothing but protein. The two proteins that we see are actin and myosin. Remember I told you, my brain says myofibril, sarcomere, and sarcomere has the myofilament in it. Why? Because the sarcomere is basically a contractile unit, the fundamental contractile unit, and you can see a sarcomere here. So you'll have a lot of proteins involved, but this large purple one going across, that's myosin. And then the little thread-like here, beads on a string-looking thing here, the orangish to me, maybe a golden yellow or something, that actually is the actin. And so if this whole thing is a sarcomere here, and we can figure it out by the zigzag boundaries, then they're made up of myofilaments. That's the reason why I keep it in my head as sarcomeres are made of myofilaments. Okay? And guess what, folks? This actually gets to create that striated appearance on skeletal muscle. And why it's such this repeating pattern that we saw that was really distinctive on skeletal muscle tissue in the light microscope. Because all of them, we have a little contractile unit here, here, probably another one over here. It's what all those myofibers are made of. And when you take a cross section of that, you're being able to see that complete crosswise pattern. Okay, folks, so again, we're priming our brain, we're getting some stuff, we're going to go into more details of the microscopic forms of the sarcomere and how it actually shortens. So first, let's get into a little example here, and then we'll move on. I'm going to focus on number 6. It says, arrange the following from superficial to deep. So superficial is something you probably can feel, whereas deep, you would have to go underneath the superficial layer in order to feel it, okay? So for example, if I'm just touching my muscle on my forearm here, I'm like, okay, I can touch the muscle here. And then if I need to touch the muscles underneath it, that's me going deeper and deeper and deeper. So when they're telling you this and they're saying, place the correct letter in each blank, let's see how we can connect it together. We have myofilaments, epimysium, perimysium, endomysium, and sarcolemma. Okay? Now what they're asking is, which one is going to be more superficial, like larger? And then we need to go all the way to smaller. So I see superficial here, then I see a random fascicle in the middle, then I see myofibrils, and then I see deep. Okay, so let's go ahead and put these in order. Well, again, I can just look at things here. Epimysium has to do with the whole muscle. Perimysium has to do with fascicles. And when you're actually studying and you're basically doing all these worksheets and stuff, make sure you use your notes and the videos and everything you can, or whoever else you're listening to, because you don't just have to listen to my videos, right? There's so many amazing people out there on YouTube that you can actually listen to who might have a better vibe that you're looking for. But I want to make sure that you write things down. Write your notes down, because this is the time to practice. Some folks will be like, oh, it's all in my head. Don't keep it stored in your head. Practice, practice, practice. Become more active, okay? That's the reason why those active reading guides are so cool, because it forces you to kind of look for information versus just memorizing a bunch of paragraphs that you're reading. All right, folks? So you got your muscle, you got your fascicles. Your endomysium is going to be dealing with muscle cells. Your sarcolemma is going to be the plasma membrane surrounding the myofibrils. And then myofilaments are going to be within these sarcomeres. Okay? So we got to start off with things that are really large, and we're going to something that is deep, really, really within, right, the muscle itself. So of course, if we're talking about muscle to start off with, the things we can touch, then it's definitely going to be letter B, because that's always focused on the full muscle. And muscle is made of what? Bundles of fascicles that are surrounded by what? The perimysium. So that's going to allow us to have letter C. And as you can see, we have our fascicles that are next. So we had our epimysium, which is the muscle. Then we had our perimysium, which is surrounding the fascicle. Now let's continue on. What's next? The fascicles are made of bundles of what? Muscle cells or muscle fibers, right? And they're surrounded by what? Endomysium. That allows us to get rid of the muscle cells as well. And then what else do we have left? We have sarcolemma and we have myofilaments. The question you have to ask yourself is, will I be able to reach the sarcolemma first, or will I reach the myofilaments first? Well, the sarcolemma is the plasma membrane of the myofibrils. The myofibrils are made of sarcomeres that are made with myofilaments. So the sarcomeres and the myofilaments would be the last that we would have here, because it's going to be the smallest piece or the smallest fundamental unit, right? Whereas the sarcolemma will come before that because it's surrounding that myofibril. Let me erase that because I know I was supposed to put myofilaments in too. All right, we're back. There we go. So this means that we're talking epimysium for the whole muscle, perimysium surrounding the fascicle, a fascicle itself, and then endomysium surrounding the muscle cell. Then we're going to have the sarcolemma surrounding the myofibril, which is, of course, bundled in the actual muscles themselves. Then we have the myofibril, and then we're going to have the myofilaments, which myofibrils are made of because they're made of these repeated units of sarcomeres. So now we have everything that we're looking at. All right, folks, I'll see you in the next video where we actually get to learn about how contraction works with these sarcomeres, using the sliding filament theory.